Awesome. Okay, so deep breath, finally made it. Woo, yay, yay cars. Just want to introduce this next section where it gets, a little hairier. The depths, we're kind of seeing the same types of mechanisms and pathways, and this is where it can get confusing. So I wanted to start and kind of put a little bit of this out there, just so that you know where you're going and what all you'll need to do to get there. Yeah, it's terrible starting. You can tell I'm still all verklempt from my car issues. Anyway, so what we've looked at is fatty acid oxidation, where we're breaking it down. Now we're going to look at how we build it up. And besides our fatty acids, we're going to look at various other lipid bodies in there, like the eicosanoids. We'll see that those can be used to generate a lot of other biomolecules in there. Was it our thrombins, our leukotrienes, and stuff like that. We're going to look at how these assemble from fatty acids and glycerol into triacylglycerols. So just like we break down, we're going to build up. We'll also talk about how we make cholesterol, which is a surprisingly complex series of reactions. Then we'll look at how we move around our cholesterol as well too. And this is where it gets very interesting. I actually found this when I was a student. This was probably one of the more fascinating parts. And I don't know if it was just, our instructor did a really good job of explaining this, or I was just interested in it. But we'll talk about these various lipid bodies that transport cholesterol in there. And of course, we'll talk about the regulation, which is always an interesting aspect. I don't know if you remember, there used to be these series of commercials on TV for cholesterol medications, like you get your cholesterol from your Aunt Vi and your apple pie, because about half of it roughly comes from diet, half of it is made in the body. So what that says is cholesterol plays a critical role in the body. It's just balancing it out, maintaining proper homeostasis, which is challenging with diets. Okay. So you've probably seen this slide before. If we go way back to I think it's chapter 10, we talk about lipids to fill a variety of biological functions. I know we were talking about birds here a little bit earlier, but my daughter wanted the bird. Well, my wife wanted a crazy bird feeder, and my daughter wanted to fill it up with seeds. So we get various birds out there. And if you look at why are male cardinals so red, it's because of the type of seeds they eat. Finches have yellow feathers because of what they eat. So presumably whatever's in there that the blue jays are eating are what contribute to the blue color of their fur in there. But more other than that, other than pigmentation, lipids, clearly energy storage and membrane components in there. But there's a lot of cofactors and signaling molecules and even transporters that are lipid based in there. And we'll see some examples of those here. All right, so let's talk about catabolism. All right, we're going to produce acetyl-CoA and produce reducing power, NADH. So this is what's on the test coming up next week. We know we're breaking down. I always think of being a cat for catabolism, scratching things in little pieces. We're making acetyl-CoA, we're making NADH in there. The opposite pathway is the anabolism, which is going to require acetyl-CoA and malonyl-CoA as well. And we're going to require the reducing power of NADPH. Okay, so we're going to get our reducing power from NADPH, and we're going to start building on these acetyl-CoAs to make these longer chains in here. As far as we're concerned, this all takes place in the cytosol. Where does our fatty acid oxidation take place? Yep, mitochondria. There we go. That's all good. Yeah. So what we've done is we've separated these two opposite processes. All right. So we don't have a futile cycle going on. So we break it down in the mitochondria and we build it up in the cytosol. This is I want to make jokes, but they're not funny. They never are. All right. So if we look here at our animal cells, and I always like this yeast. And it's like, what is yeast? Is it a plant? Is it an animal? But we can see our cytosol, our NADPH production in there. Remember long ago, it seems like we talked about the pentose phosphate pathway and our malic enzyme. That's where we're getting our NADPH from. When our NADPH to NADP plus ratio is high, that is again another trigger of a high energy state for the cell. So we can use some of that reducing power to synthesize our fatty acids and our cholesterols. Okay. So isoprenoids and sterol synthesis, which we'll see coming up in a little bit. The endoplasmic reticulum is where we see phospholipid synthesis, sterol synthesis in the later stages. So we start in the cytosol for sterols and move to the ER. And then once we have our fatty acids made for our elongation or our desaturation, that will occur in the endoplasmic reticulum. And in the mitochondria, we can also see some fatty acid elongation in there as well too. But really, it's going to be our fatty acid oxidation. It's a weird one. We're not gonna sweat that last point. Okay. So how do we build fatty acids? Okay. Well, just like when we oxidize them, we cleave off 2 carbons at a time, make our acetyl-CoA. We're basically going to build them 2 carbons at a time with 1 acetate unit. However, we aren't just going to take the acetate unit from acetyl-CoA and pass it on to another one to go 4x4. We're actually going to use malonyl-CoA, which is our 3 carbon unit here. And we're going to take 2 of these carbons and pass them on. Actually, each pass involves a reduction of a carbonyl carbon to a methylene carbon. All right. So we're going to see how this works here, but we need malonyl-CoA instead of just acetyl-CoA. So the fatty acid synthesis occurs in all compartments where NADPH levels are very high, typically the cytosol. Where does our NADPH come from? Pentose phosphate pathway. In plants, they can make it from photosynthesis, but we're not going to worry about that. So remember that our NADPH comes from the pentose phosphate pathway, and you're good for your sources in there. For adipocytes, this is malate being converted to pyruvate, and in hepatocytes, it's glucose 6-phosphate to alternate fates of glucose. Okay, and so here you see the malate being converted to pyruvate in the glucose 6-phosphate to ribulose 5-phosphate. Okay, so just a kind of reminder of sort of what we've seen in there. Okay, so how do we get our malonyl-CoA? Well, we're going to combine an acetyl-CoA with a bicarbonate. So essentially, we're going to have this single carbon transfer coming from bicarbonate onto our acetyl-CoA, right? And I always think that this one should be a fairly easy enzyme to remember. Acetyl-CoA carboxylase, abbreviated ACC. Here we are in the middle of North Carolina, and ACC country for basketball, football, whatever in there. So ACC is our acetyl-CoA carboxylase. This is a multi-subunit enzyme. There is a biotin in there, which is covalently linked to the enzyme, and that's going to carry the carbon dioxide molecule in there. And all 3 subunits are on 1 polypeptide chain. So it's a 3 subunit enzyme, but it's 1 long strand in there. Okay. So how does this work? We've seen some carboxylations before, our pyruvate carboxylase and propionyl-CoA carboxylase. We've seen in previous examples of this, but we see our biotin carrier protein. We have a biotin carboxylase area where we attach that carbon dioxide from bicarbonate to the biotin. And then that biotin arm is going to swing over into the trans carboxylase, which is going to transfer the carboxy group onto the acetyl-CoA. All right. So part 2, that arm swings over here. Our acetyl-CoA binds into the trans carboxylase section, and we transfer that carbon dioxide to the acetyl-CoA to make malonyl-CoA. Okay. I was going to say, for once, we aren't worrying too much about the mechanism, right? Because now we're in there. Okay. All right. So that's our ACC. That's our starting producer of malonyl-CoA. Okay. When we actually want to synthesize full fatty acids, This is catalyzed by the nicely named fatty acid synthase. All right. So this is a 4-step sequence that is basically going to build up the fatty acid chain to elongate it in there. So we got 4 steps in here. And then by that point, we've added, I believe, 2 carbons. Yes, 2 carbons at each step. And then we'll repeat and add 2 more, repeat, add 2 more, repeat, add 2 more. So just like fatty acid oxidation, where we cut off, we have a process where we cut off 2 carbons, and then we repeat and cut off 2 more, repeat, cut off 2 more. We're kind of doing the opposite thing here. I will say, unlike glycolysis and gluconeogenesis, completely different enzymes involved all around here. So there's not like a reversible section here, which is why we have separated these to the cytosol and the mitochondria. Okay. I should say uses. We're going to use a couple enzyme-bound sulfhydryl groups as the activating groups in here. There are variations of fatty acid synthase as well too. We're primarily going to talk about fatty acid synthase 1, but there's a fatty acid synthase 2 for plants and bacteria that does essentially the same thing, just in a slightly different manner. Fatty acid synthase 1 is, again, a big single polypeptide chain, one long enzyme in here. Fatty acid synthase 2 actually has separate chains in here that can diffuse across. Fatty acid synthase 1 only makes palmitate, right? So this is where intellectually I look at, well, yeah, we're humans, we're like higher organisms, right? And we only make palmitate? Yes, we only make palmitate. Fatty acid synthase 2 can make a whole bunch of different fatty acids. They can be saturated, unsaturated, branched. They can have all sorts of different lengths in there. Yeah, so there's a lot more variety that comes from fatty acid synthase 2. All right, but we make palmitate, and then later on we can do stuff to palmitate. Okay, so we can, but the carbon 15 and carbon 16 are from the acetyl-CoA, and they're kind of used to prime the reaction of our fatty acid synthase. Fatty acid synthase 2 only in plants and bacteria. Here we have our various subunits, and actually I should have looked at this ahead of time. Come on, there we go. We have a key down here for what those little subunits are. There is a ketoacyl synthase in there for the KS, malonyl acetyl transferase for MAT. There's a beta hydroxy dehydratase for the DH in there, so that's a dehydratase where we're pulling off our hydrogen there. Enol ACP reductase for ER. I always look at that and I think, endoplasmic reticulum. No. So all these are, just various fatty acids that are going through different reactions. And the key to this picture is we show that there's a lot of places where these reactions occur on fatty acid synthase type 1. So theoretically, if I were mean, I would ask you guys a question. What happens if I come up with an inhibitor for the KR position, the KR site on fatty acid synthase 1, and you'd say, well, we'd end up, going through this process, whatever this process is, and then we, probably get a backlog of whatever, maybe that enol hydratase, beta enol something in there, but we get an excess, an accumulation of that, which could lead to some type of disease state, and we'll probably see some disease states like that. Yay, good times, right? Okay, I want to make sure I'm not going too fast here. Oh, okay, good. Yeah, this is you see, this is a really, really long chapter, so I'm not going to go too far into it. So fatty acid synthesis, our overall goal is to add 2 carbon units to a growing chain and reduce it. Okay, so add these carbon units, reduce. So we have a 4-step process. There's a condensation, reduction, dehydration, and another reduction. Okay, so the condensation is where we add the acetate on there. It's a condensation reaction, so you can see we're going to be removing water. We're going to reduce this carbonyl to a hydroxyl group, so we'll have a C double bond O. There's a hawk flying around outside the windows up there.
Really?
Yeah. Well, it just swooped by twice, so I was like, man, okay, I think I like this classroom a little bit better. You didn't see that in the old building. Anyway, we're reducing our carbonyl group to a hydroxyl group, and then we're going to dehydrate the alcohol to a trans alkene. So we're going to make that double bond, and then we're going to reduce that double bond to an alkane. Okay. So essentially we are removing oxygens in there. We're reducing our step. Okay. So our prep, we get malonyl CoA and an acetyl CoA bound onto our fatty acid synthase 1, and we're going to lose one of those CoAs. Okay, so they're going to bind through a thioester, one of those sulfhydryl groups in there, typically a cysteine, and this will activate the acyl group and get it ready for reaction. So step 1, we're going to condense the 2 carbons from the acetyl group to 2 carbons from the malonyl group. Because malonyl has 3 carbons, we're releasing one of them as carbon dioxide. Right? And this gives us this beta keto intermediate. All right.
Wait, quick question.
Yes.
Why is it considered activated acetate? What makes it activated again?
So when we are bound with the sulfhydryl group, we have, I guess, an electron withdrawing area in there. You got to go back to a little bit more organic chemistry than I remember off the top of my head, but it's kind of priming it to make it a more reactive group. So the acetyl group by itself is kind of content to sit on the CoA. We need to do something to it to make it more thermodynamically favorable to interact with the malonyl group. So first step, this condensation. So we end up making this acetoacetyl ACP, right? Which always makes me think of it must be lunchtime because the Mexican restaurants around here that have the ACP. What is it? This chicken and a cheese sauce over the nice Spanish rice. That was my go-to lunch for too long. Such a heavy lunch there. Anyway. Yeah, exactly. That was nap time. And going back to organic chemistry, it's like, look, it's an example of the Claisen condensation. So just like, it's a Claisen. Right. And this is our beta ketoacyl ACP synthase, or our KS region of that fatty acid synthase in there. The neat thing, the reason that this becomes favorable is that condensation is coupled to the decarboxylation, and that allows this to be energetically favorable. Okay. All right. So here's our malonyl group, bound to our acetyl CoA in there. Here is our acetyl group that is been transferred to the sulfhydryl, so that our sulfur becomes a good leaving group, so we can have this attack from our second carbon in the malonyl group, and that gives us this nice lovely little condensation here, and this becomes our alpha and then our beta keto acid. Okay, our second reaction, we reduce that so that our NADPH comes in and gives us some nice reducing power. We're going to pick up a proton as well too. That's why our NADPH becomes NAD plus, and as we add that proton to the carbon in there, we end up with a lovely hydroxyl group. Okay. And just in this drawing, both the malonyl CoA and the acetyl CoA have already been attached to the complex through those thioester linkages in there. Okay. So we reduce this beta keto intermediate to an alcohol. This is our ketoacyl reductase, so Kr in there. Our third step, we're going to dehydrate it. All right, I'm going to stick it in a dehydrator and make some jerky, lipid jerky. Oh, that sounds terrible. But yeah, so we're going to make that lovely double bond in there. This will be a trans alkene. Okay, so clearly now all we need to do is reduce across that double bond, add a couple more protons in there. So we're going to use another NADPH and give us a saturated alkane. Okay. All right. And this is our enoyl reductase, or ER. So Kr is first and then ER. Okay. All right. And that's what we get. So now what we've done is we've managed to take this bound malonyl CoA, lose a carbon as carbon dioxide, but add an acyl group to it, an aceto group. So now we have this acetoacetyl CoA, and this is saturated. And we can repeat this reaction again. Okay. And we will do that, and we'll continue to add these carbon units in there until they grow longer and longer and longer. For more was it, a total of 7 steps in there, so that we have a 16 chain saturated fatty acid, and this is our palmitate. Okay. So fatty acid synthase 1 makes one product, and it makes it well. We're going to make palmitate, saturated 16 carbon fatty acid. Okay. All right. I tell you. All right, so let's talk a little bit about, how this kind of works out physiologically. There's this acyl carrier protein group in there that serves as a shuttle in the fatty acid synthase. So just like how we saw biotin can pick up a carbon group and move it from one place to the other, we go back to our ACC, we're adding that one carbon to the acetyl-CoA to make malonyl-CoA. Our acyl carrier protein is going to be our shuttle for the fatty acid synthesis, and it can have this prosthetic group in here. You can see this lovely little chain in here with the pantothenic acids. I think we've talked about the pantothenic acids a little bit before, but the key is there is a sulfhydryl group at the end, and that's where we can attach our fatty acids. And what our acyl carrier protein does is it basically shuttles the chain as it grows in there to another. Once it gets bigger, it gets too big for that site, so we shuttle it to another site so we can extend it later on. And that's why we saw, this starts off in the cytosol, moves to the endoplasmic reticulum as we get longer. Charging ACP FAS1 with the acyl groups activates them. So we have those sulfhydryl groups, we got to attach something to them, and that's going to make them more reactive and allow them to have an energetically favorable reaction in there. We can calculate thermo. No, we're not going to do that. Okay. But once we've got those both activated, then they're reactive and we can transfer the acetyl group to the ACP. Okay. And of course, our ACP sulfhydryl group gets recharged with malonyl from malonyl. Okay. Once we have our long thing. So I kind of like this picture because I think this kind of shows kind of our steps that as we go through this reaction, we bind our stuff. We see, here's our keto reductase in there with our malonyl-CoA, and we can see this kind of interact to give us this binding. This kind of, I don't know. I think this is one of those things where you kind of have to stare at it a little bit and figure out what it's telling you. But we have our different regions in here for our initial setup and our 4 different reactions: our condensation, reduction, dehydration, and second reduction. Okay. And that's going to be a process I'm absolutely going to ask about for fatty acid synthase. What are the reactions that occur in there? And you don't need to know beta keto acyl ACP, whatever, beta hydroxy acyl, those terms in there. I just want to know that as we go through this process of adding a 2-carbon unit, we start with a condensation to add the 2-carbon unit there, then a reduction, then a dehydration, and then a second reduction to make the alkane to get our growing chain. We do that several times until we get palmitate. We pass these through, we get our condensation, we go into the next step, our dehydration, our second reduction, and then our translocation to the next one so that we're ready to recharge it and go again. It's like, how can we draw out a 4-step reaction into 6 steps? Basically, it's transferring our products and then recharging them, refreshing them. Okay. These are the enzymes. I'm not going to make you memorize these. But what I want you to know again is condensation, reduction, dehydration, reduction, and then of course transferring the products away and recharging your starting components. Okay. So the last thing I want to talk about for today is just the stoichiometry of this reaction. We're basically going to take 7 acetyl groups and we're going to carboxylate them to make 7 malonyl-CoAs. So we're going to basically suck up 7 carbon dioxides to do this as well. This is going to require ATP for our acetyl-CoA carboxylase to make our 7 malonyl-CoAs. And then we're going to have 7 cycles of condensation reduction, dehydration reduction in there. We add all that together. We'll need 14 NADPHs because there are 2 reductions for each pass through there. And we're going to suck up a lot of protons as well too. We'll make palmitate and we get back, 7 carbon dioxides. So it's not like we're really using up that carbon dioxide. We're just putting it back in a later step. And we get our CoAs and a bunch of water that comes out of this. Okay. All right, but we end up with palmitate for these 7 passes in there. Reminds me of like when I was a kid, all the songs that had 7s in them, camp songs or stuff. Okay. And I think since this is an abbreviated class, this is probably a good stopping point for today. So I'm going to stop the recording and stop the sharing. Stop it all.